So, good morning. Today I am going to talk about the bite marks and lip prints. To begin in forensic odontology, what are bite marks? Bite marks are the marks which are caused by teeth alone or in combination of teeth with other parts of the mouth. So, bite marks are the marks which are caused by teeth alone or by teeth in combination with other mouth parts is known as bite marks. Now, these bite marks actually represent a very primitive form of self defense or violent attack. It represents a very primitive form of self defense or violent attack. Bite marks can be either due to human interference or by animal interference. Human bite marks are usually broad, U-shaped and circular or oval in nature. They are broad, U-shaped and circular or oval in nature, whilst animal bite marks are narrow, deep and V-shaped. Animal bite marks are narrow, deep and V-shaped. There are certain other differences regarding human and animal bite marks also. That is, in case of human bite marks, your central incisor imprint, the incisal edge imprint, is broad, whilst the lateral incisal edge imprint is narrow. The central incisal edge imprint is broad and the lateral incisal edge imprint is narrow. Whilst in case of animal bite marks, the central incisal edge imprint is narrow and the lateral incisal edge imprint is broad. Along with that, there is a deep indentation or impregnation pertaining to the canine tooth. These are the shape differences and the articulation differences pertaining to human and animal bite marks. Another thing to note is that the specific areas in which bite marks are taking place. For human bite marks, especially pertaining to the sexual abuse and child abuse, the bite marks usually take place in breast, abdomen, legs, thighs, shoulders. I repeat, for human bite marks, the preferred sites are breast, abdomen, legs, thighs, and shoulders. For the animal bite marks, the preferred sites are exposed skin surfaces and the extremities. The exposed skin surfaces and the extremities. With all this, we are going to proceed with the classification of the bite marks. According to McDonald, it can be of three types. First is your tooth pressure marks. The bite marks produced as a result of direct pressure from the tooth surfaces are known as tooth pressure marks, such as the incisal edge of incisors can lead to formation of a rectangular bite mark, which is a tooth pressure mark. The occlusal surface of posterior tooth like molars can produce a bite mark, which is also a tooth pressure mark. So owing to direct pressure from the tooth, the bite mark which is produced is known as tooth pressure marks. Second comes is your tongue pressure marks. Suppose a victim has been bitten by an assailant or an attacker, and a large amount of tissue of that victim is within the mouth of the attacker. It is being thrust upon by a large force within the tongue. And what are the structures against which it is being thrust upon by the tongue? It is mostly thrust upon against the palatal rugae area and also against the alveolar process, particularly the lingual mucosa pertaining to the lower arch. It is thrust upon against the palatal rugae area and the lingual mucosa pertaining to the alveolar arch. Now, when the victim's tissue is being thrust upon by the assailant within the oral cavity, the imprints of these areas, such as the palatal rugae or the alveolar ridge, are imprinted on the victim's tissue which is within the assailant's mouth, and this is known as your tongue pressure marks. Third is your tooth scrape marks. So, when the tooth of the attacker scrapes against the tissues of the victim or the tissue surfaces of the victim producing an abrasion, these are known as tooth scrape marks. So, it is of three types. According to McDonald, the bite marks are of three types. One is tooth pressure marks, second is your tongue pressure marks, and third is your tooth scrape marks. I repeat, tooth pressure marks, tongue pressure marks, and tooth scrape marks. The attack can be of various types. Now, the bite marks can be of varied types, such as your abrasion, contusion, laceration, trauma, etc. And how do you usually identify the bite mark? A bite mark can be identified by a host of features, such as at first there is gross features. What are the gross features? A bite mark can be circular or elliptical in nature. Circular as in case of human bite marks, narrow and elliptical as in case of your animal bite marks. And in the center of the circular or elliptical bite marks, there is a area of ecchymosis which is produced due to the negative pressure of suction within the oral cavity. After the gross features, there are certain class features. What are the class features? It is specific features pertaining to the bite marks, such as owing to the incisal edge of a tooth, a rectangular bite mark will be produced. Owing to the canine tooth, a triangular bite mark will be produced, and owing to premolar tooth, sharp and pointed bite mark will be produced. I repeat. Owing to the incisors, a rectangular bite mark will be produced. Owing to the canines, a pointed and triangular bite mark will be produced, and owing to the premolars, a sharp and pointed bite mark will be produced. And after this gross and class features, there comes individual features. What are the individual features? This comes any particular features, but especially with respect to the attacker's mouth, such as presence of any fractures, any restorations, root canal treatments, crowns or veneers, etc., through which the attacker can be identified later on. Now, what are the procedures through which these bite marks can be corroborated, which are on the tissue surfaces on the victim? They can be on the tissue surfaces of the victim, on food items, or on other substances. How can they be corroborated later? If it is on the tissue surfaces of the victim, many methods are there by corroborating the bite marks on the victim, and by comparing the morphometric, craniofacial, and even histomorphometric features of the attacker's tooth. These are done by a myriad of processes, such as by thorough photography, by taking impression through a material such as your silicone. So by thorough photography, by taking an thorough impression by a material such as your silicone. And third is by producing a proper cast, which is giving a replica of the bite mark on the body of the victim and a replica of the corresponding tooth surface areas of the assailant. So these are the points to note. Now, we must be significant in that the bite marks should ideally be carefully examined. It can be an indicator of sexual abuse, such as the bite marks in the areas of breast, abdomen, necks, shoulders, etc. in case of humans and adults. And in case of children, those on genitalia and extremes of body parts can be an example of child abuse. So bite marks can help to distinguish sexual abuse, child abuse, or theft. These are very significant points and pertinent points which should be kept in your mind. Now, certain other factors. Now, what are the pattern of bite marks of fish? Now, fish are usually polyphyodont in nature, meaning they have multiple series of teeth, and as soon as one series of teeth are exfoliated, it is being replaced by another series of teeth. So if the fish bites a particular person, there will be indentations of multiple teeth as the polyphyodont in nature. For reptiles, it is a very interesting scenario. In case of your non-venomous snakes involving the maxillary arch, they usually tend to possess two layers of tooth. So in non-venomous tooth, in maxillary arch, two layers of teeth are present. Whilst in case of venomous snakes, in maxillary arch, only a single layer of teeth is present. Palatal to this single layer of teeth, there are large and long poison fangs through which poison is delivered. So if a venomous snake bites or indents a person, the indentation will be quite different from that of the non-venomous snakes. Now, with this, we continue with the bite marks. Bite mark identification can be of three types. If the victim and the attacker data completely match with each other, it confirms identification. If most of the data pertaining to the casts, photographs, and impression materials and other examination thorough procedures of the victim match with that of the attacker, then it is a probable identification. And if most of the data pertaining to the victim and the attacker, which I have mentioned previously, do not mostly match with each other, it is a possible identification. So these are the types of identification of bite marks. Now with bite marks completed, we will now shift our focus to lip prints. What are lip prints? There are numerous sulcus and grooves in the lips. The imprints which they produce is known as lip prints. There are numerous sulcus and grooves in the lips. The imprints which they produce are known as lip prints. And the study of lip prints is known as cheilooscopy. The classification of lip prints have been made by a scientist known as Sushi Hashi. The classification is as follows. First is type 1. Type 1 lip prints are clear-cut vertical grooves which run across the entire lip. Clear-cut vertical grooves which run across the entire lip. Then is 1 prime. These are clear-cut vertical grooves, but they do not span across the entire lip. Type 2 is your branched. Type 2 is your branched. Type 3 is your intersected. Type 4 is your reticular. And type 5 is your morphologically uncategorized. This cannot be attributed to any type of known morphology. So I repeat the classification. Type 1 is your clear-cut vertical grooves which span across the entire lip. Type 1 prime is your clear-cut vertical grooves but they do not span across the entire lip. Type 2 is your branched. Type 3 is your intersected, type 4 is your reticular, and type 5 is your morphologically uncategorized. They cannot be attributed to any type of known morphological parameters of lip prints. Now, how lip prints can be identified? Usually, thin film or strip is taken having magnetic or alum powders in it. A thin strip or film having magnetic or alum powders in it are taken, and it is pressed against the surface of the glass from which perhaps the assailant or the attacker has taken a drink, such as from the edge of a glass, etc. And then it is thoroughly examined and visualized. By this method, a lip print can be identified. What are the disadvantages of the lip prints? It is an anatomical parameter which is susceptible to change due to environmental and other external factors throughout the life. It is a anatomical parameter which is susceptible to change as a result of environmental, external trauma or other types of alterations throughout the human life. It is more susceptible to change than other parameters such as your palatal rugae, fingerprints or dentition. So this is its disadvantage. Now after this we'll going to talk in very brief regarding other forensic parameters such as your fingerprints. Fingerprints is a biometric aspect, and by assessing the arches, ridges, loops or holes, a fingerprint can be identified. Now with the fingerprints we go on to forensic serology. How you can identify blood and saliva. These are the two most important aspects. For identification of a blood, suppose a murderer has murdered someone and there is spillage of blood in the accidental spot. However, he or she has mopped the floor in such a manner that the blood is not visualized but the stains are not there. How would you identify that blood? Now there is a particular method. There is a spray called luminol spray. Okay, you spray the purported area of the spillage of the blood with the luminol spray, and after you spray it you visualize it under ultraviolet light. If the luminol sprayed area under ultraviolet light fluoresces green in color, then you can be almost sure that blood spillage was on that area. Okay, another method for determining the presence of blood stain is your Kastle-Myers test. How is it done? To the suspected area of blood spillage or blood stain, a drop of phenolphthalein is added, followed by a drop of hydrogen peroxide. So in Kastle-Myers test, in the area of suspected blood stain, a drop of phenolphthalein is added, followed by a drop of hydrogen peroxide. If the color turns pink, then you can be sure that there was blood spillage in that area. And another significant aspect is the identification of saliva. How can you identify saliva? This can be done by alpha amylase test. Now, within saliva, an enzyme called alpha amylase is there. The purpose of alpha amylase is to break down starch into smaller oligosaccharides. How this alpha amylase test is being performed? First, we take a petri dish. Within the petri dish, we take a gel-like substance which is composed of starch-like material. We take a gel-like substance comprised of carbohydrate and starch-like material, to which we at first add iodine. As iodine is added to the starch, it immediately becomes blue in color. To this, salivary component or concentrates of saliva are added. Now this saliva is containing the alpha amylase enzyme. As soon as the alpha amylase comes in contact with the starch, it breaks down the starch into smaller oligosaccharide residues. And as a result, due to the starch breakdown, the blue coloration which was being caused by the iodine, it disappears immediately. So by this alpha amylase test, we can detect saliva forensically. Now, with fingerprint and other methods taken, we'll briefly talk about how will you identify iris and retina by forensic methods. Now, retina is an anatomical composite structure in the posterior compartment of the eye. It has a very rich and articulate microvascular capillary supply and very rich neural innervation. I repeat, it has a very rich microvascular capillary supply and a very rich innervation. A very fascinating point to note is here that the rich microcapillary supply pertaining to each retina of a person is very unique to an individual. The capillary network within the retina is unique to an individual. It does not change until and unless the individual is affected by grave disease such as your diabetic retinopathy, by glaucoma or neurodegenerative disorders. Otherwise, the microcapillary pattern within the retina remains constant throughout the life. It will remain as it is. How can you detect the pattern of this microcapillary within the retina? By doing angiography or Doppler methods, by ultrasound guided MRI, and sometimes by confocal fluorescence microscopic method. Now, what is iris? Iris is also another important part of biometric detection, such as in case of your phone face unlock procedures. Iris is the structure which is surrounding the pupil of the eye, and it is surrounding the pupil of the eye. And what is its function? Its function is to control the amount of light which is entering the eye. So it acts as an aperture. Now, the iris surrounding the pupil is usually of brown color, but it can also be of green, blue, and even purple or pink color. The iris has a very complex and sophisticated individualistic pattern which can be identified by biophysics algorithms, and through which the iris pattern can be individually specifically identified. So with this, we complete with the forensics. Thank you for your patience.